In this video, continuation of cultivation of iris, explant fragment of tissue can be grown as explant. Adenoid explant are used for explant. So in this video, we want to look at cell culture, cell line preparation. First, look at preparation of cell line. Basically, tissue are taken, they are digested, they are dissociated into individual cells. That is first thing that they want to do so. Take the tissue, then dissociated into individual cell. We will take tissue, then we will dissociated into a individual cells. So how will you do this? By using some proteolytic enzyme. That enzyme are trypsin and collagenase. Trypsin and collagenase. Then you will do after digesting it with this enzyme, then mechanical shaking, then they will dissociated into individual cells. Then the cells are washed, counted, suspended in a viral growth medium. So you are taking these cells into a viral growth medium. For first washing these cells, counting the cell, then suspending them in a viral growth medium which containing balanced salt solution. They will be essential amino acids, vitamins, etc. The growth medium containing, what are the things it containing? Essential amino acid, vitamin, salt, glucose. Some supplemented cough serum, then antibiotics. Even antibiotics, we don't want to cultivate bacterium. So you will use a buffer to maintaining pH 7.2 to 7.4. So here we are using buffer bicarbonate and phenol red, that is added as a pH indicator. So there are using phenyl red as a pH indicator. So now moving to tissue culture flask. This one is the flask, tissue culture flask. So this is the preparation, that tissue culture flask viral growth medium containing cells is suspended in a tissue culture flask. Then monolayer sheet formation. First of all preparing cell line first took the tissue, digested into individual cells. Then took viral medium, add a lot of glucose, amino acid, vitamins, fetal calf serum, buffered with a bicarbonate to maintaining the pH, added phenyl red indicator to it. Then we look tissue culture flask and suspended to a viral growth medium into that tissue culture flask. Now we are going to monolayer sheet. Monolayer sheet formation. In this monolayer sheet formation is in this, what happened on incubation, the cells adhere to that glass surface of the flask and they divided to form a monolayer sheet of cells within a week, covering the floor of tissue culture flask. So basically all incubation this happen, they are saying it will become a monolayer of that cell or all one layer. Incubation, this tissue culture flask are incubated horizontally. So this flask is in horizontal. horizontally in the presence of carbon dioxide. So in the presence of carbon dioxide, so definitely that lid is closed. You can see here carbon dioxide either as a stationary culture or roller drum culture. Rolling of that culture bottle in roller drum provided better aeration, which is useful for isolation of fastidious virus like rotavirus. So that was about incubation. So here we are incubated, then we become monolayer in the presence of carbon dioxide horizontally, either stationary or roller drum. So basically the roller will provide it a little more aeration, which is useful for isolation of fastidious viruses like rotavirus. So let's go to type of cell line. What are the types of cell line? Primary cell line, secondary or diploid cell line, third one is continuous. Continuous means it will be cancer cell, they will be immortal and dividing. So this will be continuous cell line. So cell line culture can be classified based on their origin. Chromosome characteristic, maximum number of cell division that can undergo. So the primary cell line are derived from normal cells. Normal cell freshly taken from organ or culture. So these the freshly taken, these are normal. They are capable of very limited growth. Very limited growth. They will have any capacity of these primary cell line would be very limited growth, and they can divided only 5 to 10 times. They divided only 5 to 10 times. 5 to 10 times they can grow. These are normal tissue. They maintain diploid karyosome. They maintaining as a diploid karyosome. There are useful for primary isolation as well as growth of virus for vaccine production. So used for primary isolation, growth of virus for vaccine production. Example, monkey kidney cell line, human amnion cell line, then chick embryo cell line. This monkey kidney cell line, which are useful for isolation of myxovirus, enterovirus, adenovirus. Myxovirus example, influenza virus. So all these are example of primary cell line. Secondary or diploid cell line. They can divided up to 10 to 50 times before they undergo death sentences. So 10 to 50 times. How many times would primary cell line divided? That one 5 to 10 times. This one 10 to 50 times. Basically these are also derived from normal host cells only. They maintain diploid karyosome only. For example, here diploid cell line derived from human fibroblast. Like here there are useful for some fastidious organism. Human fibroblast cell line excellent for recovery of cytomegalovirus. So human fibroblast cell line useful for recovery of cytomegalovirus. Similarly, we have something called human embryonic lung cell line. Here that is MRC5WI38. These are the human embryonic lung cell line. These are useful for preparation of vaccines. Vaccines for rabies, chickenpox, hepatitis B, hepatitis A, then MMR vaccine. For all these can use like herpes simplex virus, cytomegalovirus, adenovirus, picoRNA virus. A lot of things you can use this for vaccines. So growth of virus, a lot of spectrum is very big here. So you can grow lot of viruses in this human embryonic lung cell strain. Next one is continuous cell line. They are actually derived from cancerous cell line. There are immortal, capable of indefinite growth. So this one, they possess haploid chromosome, altered haploid chromosome. They are cancerous cell, immortal haploid, and they are easily to maintaining serial subculture. For indefinite division. There are immortal, they are not going to die. They will die in other end. This is the reason why continuous line are most widely used in cell line. Currently all the things, organ explant, animal inoculation, all are gone, and cell line are mainly used nowadays. Cell lines are mainly used nowadays. Basically, common example, HeLa, human carcinoma of cervix cell line. They are taken cancer cells from human, using it to culture virus. So HeLa cell line of human carcinoma of cervix cell line. Second one is HEP2. This is the cell line, basically human epithelium of larynx cell line, and widely used for RSV, respiratory syncytial virus, and Herpes simplex virus. Then that we can use for HBE2 cell line, from where this is coming from larynx. This is the epithelioma from larynx cell line. Epithelioma from larynx cell line. Then KP cell line from nasopharyngeal from carcinoma. Then McCoy cell line from synovial carcinoma. Then Vero cell line from vervet monkey kidney cell line, used for rabies vaccine production. Then PHK, baby hamster kidney cell line. So this was about types of cell lines. So cultivation of virus. In this one, first one is animal inoculation, egg inoculation. First video we discussed. In this video we are discussing cell lines. In this cell line, preparation of cell line, then types of cell line. 3 type of cell line: primary cell line, secondary, and continuous cell line. Thanks for watching.